So stem cells, we call them the superheroes of the body. They're unique cells that can become any cell type in the body. And we have focused here at NYSCF on a particular type of stem cell called a pluripotent stem cell. This is a cell that can make all the different cell types in the body, from brain cells to heart cells, blood cells, lung cells, muscle cells, all of the different cells that make up our different tissues in our body. But they have one really important other characteristic, and that is that they can self-renew. They can make an unlimited number of copies of themselves if you give them the right signals. So why do we think that they're the future of medicine then? So basically, we see three really main critical applications of the way that we think stem cells are going to be critical for developing new therapies and treatments. And those are through disease research, trying to understand more about disease and how different diseases affect the cells that they impact. And then the traditional sort of more common idea of basically using those cells to create more cells to replace dead or dying tissues in the body. But they can also be used for drug discovery and trying to find new drugs to treat different types of diseases. But fundamentally, we cannot treat what we don't understand. We need to be able to look at actual cells, human cells, that are affected by these different diseases. Many of these cells are not cells that we would really have access to. I would not really be able to ask you for cells from your brain to be able to study different brain diseases. We need another way to do that. And stem cells really offer a path forward here for looking at cells and tissues that we just don't really have access to in the laboratory. So we have been employing stem cells as a tool to try to understand how our differing genetics can result in the degeneration and the different aspects of Alzheimer's that we see. And we can do that by studying stem cell derived tissues from patients with Alzheimer's in the laboratory. And it's critical because we can now make all the different types of brain cells from the stem cell. I think a really good example of how far this has come over the past decade is in ALS. We can make the motor neurons that are the type of cell that degenerates in Lou Gehrig's disease in ALS. A particular characteristic of motor neurons derived from ALS patients was that they're hyperactive, they're overactive. And so they did a drug screen to look at initially existing drugs and found this ezogabine. It goes by different names, but it's a drug that has historically been used to treat epilepsy and could quell the overactive motor neurons in a key hallmark of ALS. And they're pursuing the development of this drug, but also others that they hope will improve the outcome for people with ALS. And the flip side of that is, we hope we can find drugs that work to treat the disease, but we also want to find safe drugs. So one of the major reasons why drugs fail is that they're toxic to different cells in the body. We can make things like cardiomyocytes from the stem cells, and one of the main toxicities to many drugs is they affect the heartbeat. The cells that we make from the cardiomyocytes, the heart cells that we make from the stem cells from a person, actually beat and reflect the biology of that person. So we can know ahead of time whether or not that person might have a toxic reaction to one of the drugs that we develop. The other way that we're interested in using stem cells is basically creating new cells to replace dead or dying or malfunctioning cells that might occur in different types of disease. And we're involved in a study developing a cell therapy for an eye disease called age-related macular degeneration. And our sort of inspiration for this study is based off of a somewhat similar type of study that was run by Pete Coffey and his colleagues in London, where they started from embryonic stem cells to derive these retinal pigment epithelial cells and use those in patches to transplant into two individuals in safety trials. And in both of those individuals, they were able to actually restore some of their affected sight in the eye that was transplanted. So they went from reading very few words per minute to reading 60 to 80 words per minute, essentially restoring their ability to read from that eye.